All right, in the last section we talked about action potentials, and here we'll go into a little bit more detail with what's actually going on and kind of reiterate some of the stuff that we talked about in class there. So we said that action potentials, of course, are all or none. So you get a stimulus, and then if the action potential starts to fire, it fires just as it normally would at any other point in time. And these are going to carry the signals from that stimulus that we've received over a long period, or excuse me, over a long distance in a very short period of time. And we can generate these things because they're so fast and very high frequency. And we talked about the height that we would see if we were monitoring these on a computer is going to be the same no matter what. The intensity of the actual stimulus is going to be the frequency with which we see these action potentials. And we have sodium and potassium voltage gated ion channels that are responsible for the production of the action potential, and both of these things are contributing really to the depolarization and then the repolarization of the membrane. And you have the sodium channels that are going to open first, and then after that you're going to have the potassium channels that are going to open, and they're going to kind of restore that membrane potential along with the help of the sodium potassium pump. Okay, so the sodium channels have two gates. They have an activation gate and an inactivation gate, and both of them have to be open in order for sodium to kind of flow through. And at the resting potential, the activation gate is closed, but the inactivation gate is open. And depolarization, once we start to kind of receive a stimulus, we start to depolarize the cell up towards threshold, the activation gate starts to open up, and that inactivation gate starts to close slowly. So if you kind of look down here at the bottom, you can see that, really at rest here we're open as far as the inactivation gate, but this activation gate is closed. And then as we start to achieve stimulus now, we're going to start to open this activation gate, and now these sodium ions can kind of pass through there. Okay, and when we look at the really the rest here, we've got the potassium channel here. This is closed as well. Okay, so the activation gate is closed during this, and what's depolarization is doing is it's kind of working to open this up and let these ions start to flow through. So once we get a stimulus that kind of depolarizes this membrane, these activation gates start to open, and they allow more and more sodium to come in and bring it closer and closer towards threshold. And this, once we get to threshold, these activation gates are all going to start flying open in a positive feedback kind of mechanism, and that's what's going to contribute to this large spike in the generation of this action potential up to about positive 30 right here. Okay, right in that neighborhood, 30 millivolts. All right. So now, get here. Okay, so now we don't quite reach the equilibrium of the sodium ions, but these activation gates now, these are starting to close, and now the sodium influx is starting to stop. And these when this becomes positive up here, what we're getting is these potassium gates start to open up as well. And now these potassium gates start to open, and now you have potassium flying out of the cell. And once these positive ions start flying out of the cell, now we're starting to restore that kind of negative resting potential on the inside. And all the while you have the sodium potassium ATPase pump that's working to move the ions, 3 sodiums out, 2 sodiums in, and so forth. Okay. Because these potassium gates don't close instantaneously, we get what's known as undershoot. And that's this little kind of spot right here where you can see we're becoming more negative than we would be at rest. So here's where we would be at rest. This undershoot is because those potassium ion channels are closing slowly, and more potassium is continuing to diffuse out of the cell when those channels are open. Okay, and eventually the sodium potassium ATPase pump restores us to that resting potential, and we're ready to fire another action potential. Okay, so we talked about action potentials, and we talked about in the last segment of the lecture, we talked about how these sodium ions are moving out, and then the potassium ions start to move in, and the sodium ions are starting to move in, rather, and the potassium ions are starting to move out, and that's what's restoring really the inside. At first is positive, and then restoring this negativity on the inside with regard to the membrane potential. And this would be moving all along the axon, all along this length of the axon, and it would take a long time if it weren't for these Schwann cells that we have here. So if we look over here, we've got these white cells here that are kind of wrapped around the axon. You can see how this looks a little bit different, say, than this one up here. And what those, what that myelination does, what those Schwann cells do, is they kind of act like an insulator. So instead of this current that we're seeing, or this electrical charge, the action potential having to move at every successive spot along the axon, what we can do is we have these axons that in these nodes of Ranvier, if we look right here, we can see these nodes of Ranvier, these things, this charge is kind of jumping from spot to spot to spot in terms of the action potential. And what happens is because of the myelination that we're seeing along here, those ions can very rapidly move from the point of one action potential to the point of the next action potential. So this is termed saltatory conduction. And what happens is that action potential is able to jump from node to node to node instead of having to slowly pass through the entire neuron at every single spot in the membrane. It can jump from spot to spot to spot. And those action potentials being able to go very rapidly is what enables really us to do the things that we do on a normal basis, from the standpoint of, like, if you think about it, if someone throws something to you, all of the coordination that has to go into either catching it or getting out of the way of it or whatever happens, because of the rapid conduction of that nerve impulse, that stimulus that you're seeing. And we, if we look at certain things that we talked about in class, for instance, Lou Gehrig's disease or ALS, that results in a demyelination, or in other words, these Schwann cells start to deteriorate, and now that nerve, you don't have this saltatory conduction that we were talking about, and what you actually have is now that nerve impulse has to travel through the membrane, like you would see here, through each successive spot, so the nerve transmission is greatly slowed. So that would be kind of a way to look at what the difference between myelination and demyelination is with regard to how quickly that nerve impulse can travel. Okay? So one of the other things that we talked about too was the idea of a refractory period, the point at which you can't get any action potential traveling. And there was an absolute action potential that we discussed, and this is where these sodium channels are remaining closed and there's no stimulus at all that can trigger an action potential. And what this does is it prevents really the triggering of the another action potential, and it prevents the backflow. And we talked about some of the reasons why. So if we're looking here, you can see right here, this is the absolute refractory period. So we're looking at the point really right here, if I can draw this line down to right here. And what you've got now is these sodium channels. Once these things become activated, here's your activation gate. Once this thing opens, these sodium atoms can come flying into the cell. They're changing the membrane potential right here. It's becoming very positive as these sodium atoms come in. And then what happens is now those inactivation gates from the potassium channels start to open up. And then during this absolute refractory period now, you have this inactivation gate that you see right here, okay? And this thing is going to kind of move up into this position right here. And now it's acting kind of like a plug in a drain, and it's not allowing the sodium ions to move. And now there's no stimulus at all that can cause an action potential. And then slowly this thing, and slowly I say kind of tongue-in-cheek in that it's just a few milliseconds, but slowly now this inactivation gate starts to let go. And then what you have is this activation gate is going to kind of close to restore us to this point right over here, and now we can get another stimulus occur. Okay, so what that's doing now, if you think about it, is it's allowing this action potential. We talked about this in class. It's allowing this action potential to move in this direction only. In other words, it can't move in this direction, because what's happening is those inactivation gates are right here. These inactivation gates are plugging up the sodium channels, so we can't fire any action potential until we get back to our resting membrane potential. So really it's essentially preventing the backflow of information. We're ensuring that the action potential can only run in one direction. Okay. Then we have what's known as a relative refractory period, and this is where you can have an action potential, but the sodium gates are newly closed, and along with that, we've allowed, as we mentioned earlier, too much potassium out. So we get this undershoot occurring right here. And now it's going to take a stronger stimulus in order to generate that action potential. So this is the relative refractory period. Okay. And you can see that that's occurring from right about here to right about here once we get returned to the resting membrane potential. Okay, so that's due to the potassium ions flying out and those sodium channels not really all returning back to having their activation gates closed up. So both of these things are working to prevent the backflow of the stimulus and allowing the membrane potential to kind of reset so that we can fire another action potential. Okay, so in the next phase of this, we're going to show how these action potentials, what happens when they get to the synapse. All right, so if you got any questions, of course, write them down like usual, and we'll talk to you soon.